Is mRNA transcribed and translated? Does it have the message? Yes. All right. How about rRNA? Do we translate rRNA? No. It goes and sticks with proteins and makes our ribosomes. How about our tRNA? Do we make any protein from the tRNA itself? No. tRNA goes, and as soon as it's made, RNA is made, it folds on itself. So only mRNA. Which of the following RNAs are or is involved in translation? Is mRNA part of translation? Yeah. Is tRNA part of translation? Yes. How about rRNA? Where's rRNA? Makes the ribosome. So all three of them are part of translation. rRNA makes the ribosome. tRNA brings the amino acid and translates. mRNA has the message. So technically all three are involved. They're not all translated, but they are involved in the translation. So see the difference? Being involved in the process versus being translated. So the only one that is translated is mRNA. Common mistake for students. They tell me rRNA has codons that will affect the translation. Codon is only on mRNA. Don't ever tell me codon is on rRNA or tRNA. Codon is only on mRNA. By the way, you don't have to memorize that chart. If I ever want you guys to translate something for me, I will give you the chart. Which again, trying to, before going to mutations and all of that, let me use brain teasers. I can give you a sequence of DNA like this. Totally create random, like go on, so on and so forth. So that's that. All right, so this is my imaginary gene. Don't worry that I have start or promoter or all of that. I'm telling you this is the promoter. This is where the terminator is. For this one, give me a sequence for the mRNA. Which one of these strands would you choose? Which one would be the template? Top one. Exactly, because it's going 3 to 5. Do you care what the sequence of this is? No. Remember, mRNA is single-stranded. I have something like this and students give me a double-stranded mRNA. Like, mRNA is single-stranded. So my mRNA for this would be, not funny. You can use this as a cheat. Because remember, this is complementary to that strand. So your mRNA is going to be the other strand, just replace the T's with U. U, A, A, G, C, U, G, G, C. All right? Then I can say, all right, tell me what's the amino acid sequence for this. You go 1, 2, 3, 1, 2, 3. And then you can have the genetic codon, redundant genetic codon. I'm thinking one of them is actually a stop codon. So UAA. See, it's stop. Obviously, I won't do anything like that. Or sometimes I have something like this, and students are like, they come up to me, they're like, well, there is no AUG. Like, I made this thing up to just have you guys practice. So it's okay. You know, so this is wrong. I can't have a stop codon at the beginning. Should be there. All right? Okay. Now, another teaser for you guys. let's say I have a protein that is made up of, I know the protein has 10 amino acids. So my protein is made up of 10 amino acids. All right? How many codons are associated with this protein that has 10 amino acids? Codons. 3 codons?
Stop and start and go. Start and stop.
Okay, and?
2.
Oh, there's 10 codons.
So 2, okay, what's the,
10.
so you said 30. So we have 3, 30,
10.
10, I know what you meant.
Okay.
Yeah.
10,
Yeah.
that's it?
5. Isn't it codon
Five.
an amino acid?
Codon is 1 codon, 1 amino acid.
So 10. Oh, wow.
11.
What?
11.
Oh, because of the stop.
Start is part of that. Okay, AUG methionine. Then the other 9 codons. So AUG, 1 amino acid, plus 9 codons, that gives you 10. What about stop? Stop doesn't have an amino acid. So therefore the answer is 11. 30, I knew what you meant. You're multiplying, you're giving me nucleotides, the length of it. So the length actually will be 33 because of the stop. So, although I gotta tell you, not every protein starts with methionine. Many times they remove the methionine. But we're not going that in depth.
Does that mean the stop codon is kind of like just like a release factor kind of?
Yes,
signal
but
like
stop
that?
codon is a codon. It's still a codon. So that messenger RNA has to give me the information for the 10 amino acids, but then also has to tell the ribosome, hey, this is stop. Stop it. Don't put any more amino acids, but this is the end. So it has to start by start and stop.
But then does it fall off?
Yes, separates. So mRNA separates, the tRNA and the polypeptide chain, like they showed in the amino acid, separates, falls off, goes to whatever. And the large subunit, small subunit separate, they go right to the beginning of an mRNA and start again.
Why would a stop codon not be considered an amino acid?
Because it doesn't code for any amino acid. It just says stop. Whereas start says start, but also put methionine.
So it adds amino acids, but stop
Yes,
codon-
but stop does not add an amino acid.
Okay.
Because they start with the same thing in an amino acid, but they have to end and just say, this is it. No more amino acids are coming here. So it has to do that. Good? Trust me. Play with it at home. You will suddenly get that. There will be that moment. All right? Now, the last part on chapter 8 that I'm going to cover is mutations. Forget about everything. Mutation. Where would I see the mutation? In what molecule do I see a mutation? A mutation is a mistake that is permanent. Permanent mistakes will be where? DNA. Do I care if my RNA polymerase one time when it's making this, it makes a mistake, and instead of putting an A there, puts a G there? All right, that mRNA is worthless. It's going to make a protein that doesn't work. But the next RNA polymerase is not going to make that mistake. So that's why the process of mRNA, making mRNA for us is fast and dirty. RNA polymerase is like, seriously, you want me to edit too? I'm not gonna do that. So I'll make a mistake one time, the next time is okay. You're gonna constantly, if you saw in the image that we had for the prokaryotes, when the promoter is active, you make thousands of mRNAs. One of them is wrong, so what? The others are correct. Your template is correct. Let's say for 5 minutes, even that process is wrong. So out of 10,000 mRNAs, half of them, 5,000 are wrong. Who cares? You have 5,000 that are still good. And if the cell needs it, it will make more mRNAs. But for DNA, if DNA polymerases make a mistake and don't correct it, that's a permanent mistake. And from that point on, that cell passes on the mistake to the next generations. That is called a mutation. Okay? For a bacteria, the mutation happens, they're screwed. Okay? For us humans, if you want to think about it, humans, we're with the rest of the microbes, like even a protozoan, they make a mutation, their damage is permanent because they're single cell. For us humans, it's a little bit different. We have somatic cells. These are the cells that in your body act normal. So if a mutation happens in one of my cells, then that mutation is happening in that cell and the next generation of that cell. If it's too crazy, if it has to do with cell replication and reproduction and regulation of that, then yes, you might see a tumor or something happening. But if not, then all right, just those cells will stop functioning normally. For us humans, we have another set of cells called germ cells. Those are the cells that we have in our gonads. So they're responsible for making sperms in males and eggs in females. If a mutation happens during DNA replication in one of those stem cells or germ cells, then there's a problem because it's going to be passed on to the children with the faulty egg and all of that. So mutations, now we're going to think about single cell. Most of our microbes are single cell. So mutation has to happen in DNA replication. Then mutation will have an effect on the process of protein synthesis, gene expression. Why? Because a mutation, if it's in the sequence of an mRNA, then that mRNA is constantly going to be made wrong after the mutation. The type of mutation we're going to look at is called the point mutation. So in this class, we're just going to focus on point mutation. Point mutation means only one of the bases in DNA got changed. Mistake was involving one nucleotide. Now, one nucleotide can be substituted. So during the process of replication, instead of putting a G there, they put a C. They put an A, they put a T, whatever. It substitutes, makes a mistake, and puts the wrong base there. Or insertion. As it's going, it sees a phantom nucleotide in front of it and inserts one nucleotide. So suddenly puts here, inserts a T. Sorry, not here, so there. Instead, it puts a T. Somehow it saw an A here, it made a mistake, and inserts a T. Or the reverse, deletion. Totally bypasses this one, and it doesn't put it down. And it's not caught. So point mutation could be substitution, insertion, or deletion. Now let's look at what effects would it have on your gene expression. Mutations can result, or mutations can be neutral, harmful, or beneficial. Let's say base substitution happens. A single base is changed. if that single base substitution changes the codon, and the codon still says the same amino acid, then we're okay. So, for example, let's say so GCU GCU, all right? That codon means, let's say Lucy, I don't know. So the DNA part of it is CGA, right? So that's DNA. Normal condition, actually what normal, the term we use is wild. So this is the wild DNA, wild type, meaning originally this was the sequence of DNA. Let's say I have a mutation and DNA changes to CGT. Now my mRNA for that one becomes GCA. If that one still means leucine, who cares? That's called neutral or silent mutation. If my protein remains exactly the same, it doesn't matter. In fact, this is how we have genetic variations in us humans. Sometimes we have had these kind of mutations and the same kind of different sequences mean the same protein and the protein is functioning fine in all of us, all right? So if this happens, nothing is happening, cell is still healthy, protein is fine. We refer to this as neutral or silent mutation. It will always be if you have a base substitution. Okay. Only base substitution. Insertion and deletion, you'll see, those are horrible. So base substitution, if the information is same, we're okay. Sometimes base substitution, the information is not the same. The information can be a different amino acid. Now, the harm that it causes, it depends. That different amino acid, where is the location? How much of the 3D shape of the protein is affected by it? Sometimes, have you heard of T-cell anemia? T-cell is basically, sorry, not T-cell, sickle cell. It's a T. So sickle cell anemia is basically a T was substituted, or a base was substituted, the T was placed in there, I don't remember exactly. But it was a base substitution, one single one. So it creates a hemoglobin S. That hemoglobin, the 3D structure of it, causes the red blood cells to look like a sickled moon, rather than that donut shape. When it gets to small capillaries, that can go through capillaries that easily. Sometimes it causes tears, and the amount of oxygen that it accepts is lower. So it causes sickle cell anemia. The person is still living, but has issues, right? So the degree of harm can be different. Sometimes it's little bit of a difference. Sometimes it's too much of a difference. So that is called either missense, and in case of missense, so the example that I have, the wild type says the cat saw the rat. Now we have the cat saw the bat. The cat is still seeing, but it sees a bat. The last one is probably more detrimental. The cat was the rat, all right? So it can be, okay, not functioning that great, to not functioning at all. This protein is useless for us. So base substitution, missense means just the protein is not going to be normal. Base substitution can cause nonsense, and that's actually harmful too. Nonsense, suddenly the codon changes and puts a stop codon. somewhere in the middle, so from there on, the protein is not translated. They see it as that. So nonsense means a stop codon appeared because of base substitution. Now, deletion and additions actually create the most harmful proteins. They will always be harmful. So if I ever ask what's the most harmful mutation that you could see, is either a deletion or insertion. Why? Because they cause frame shift. So for example, in what we have, imagine if the H was removed. So a deletion happens. Remember, we go 1, 2, 3, first codon, 1, 2, 3, second codon, 1, 2, 3, third, fourth, so all of that. If I remove the H from that point on, the frame is shifted. All codons from that point on are mistaken, are wrong. So your entire protein is damaged. It's not just one amino acid, it's every amino acid from the point of mutation. That's why they are extremely harmful. I'll come back and show this at the end to you guys. Now, beneficial mutations is, all right, beneficial has to do with evolutionary process. So let's say, in case of I have sickle cell up there. Granted, sickle cell is not a great disease, but people who are carriers of sickle cell anemia, they are actually immune to malaria. Because some of their cells produce hemoglobin S. And the protozoan Plasmodium does not like hemoglobin S and does not go and infect those. So they are kind of immune. So for an evolutionary trait, this is why they believe it survived in Africa. And sickle cell, it was actually a disease. Now you can see it in other races because of the interchange of genetics. But primarily it was seen in black population, the black population. And then it changed.